I'm going to just glance over 3 different areas where we can try to use these microfabrication techniques, combine them with different kinds of chemistries and materials, and then use them for specific biological applications with the goal of trying to engineer these tissues that I mentioned. So first I'm going to talk about how we can try to regulate stem cells by using these microfabricated systems, and in a very simple ways of trying to control stem cell aggregate formation. Then I'm going to talk about 2 different ways where we try to generate kind of architecture of tissues in kind of 2 different processes. One is more of a modular or a bottom-up approach to generate tissue complexity, and basically this approach basically says that tissues are made from these functional units that repeat themselves over and over to give rise to the actual tissue. So can we try to make a functional unit and then assemble it together to generate tissue complexity? And then the other approach is more of a top-down approach, where we start off with a larger material and then carve out the kind of complexity in the microarchitecture that we want. So let's start with that. So basically, we've been very much interested in making small stuff for a long time. And this small stuff, you can make it from lots of different types of materials, lots of different types of chemistry, and what you can kind of see here are some examples of this. So these are very small structures. These are zoomed up, but if you actually look at them, you almost can't see them because they're so small. And what you can do is you can control how cells and proteins interact with these small structures. So here, you can see basically, if you look here, they both have the cross-section of these microwells. So you see these microwells here? But because in one of them, there's no, this underlying silicon wafer is not exposed, so the entire material is made from this non-adhesive chemistry. So if you put proteins here, it doesn't stick anywhere. So that is good for some applications which we're going to talk about. But if you have other surfaces, and in this case you see that the underlying substrate is exposed, so if you put proteins here, then the protein actually sticks on the surface. So what this is good for is that if you have proteins sticking, then cells actually stick to proteins, so then you can get cells stuck to particular regions and start controlling the microenvironment of cells based on these patterning approaches. So we've been using these microwells and trying to actually control stem cells and what they see. So there's a lot of computational kind of engineering approaches that one can do and to understand these systems. But what I kind of focus on is really in these small structures, fluids behave differently than what you're used to in the large macro scale world. So for example, what you see here is that fluids can flow over these small structures, but there's this is a velocity gradient, so it means that these blue things are low velocity. So basically, inside these structures, you have low velocity, and the fluids that are flowing on top skip over these, and in these little wells. So this means that if you have a little well, and you have a cell or a particle in there, then the cell or particle stays there, and then the fluid kind of passes over it. So just to show you some examples of this, this is some of this stuff. Okay, we can go here. So this is an example of this. So you see these little wells, and these little dams, and these little structures here. The cells kind of stay in here, or get stuck in here, but they don't really stay here. And what that means is that because if you flow liquids here, then inside these little wells you have low shear stress. The fluid kind of skips over it, so you have the cell in there, and the cell doesn't get washed away. So what you can use this for is to try to control this location of cells on these surfaces, and try to control how they can be used for different applications. So for us, we've been interested in stem cells and trying to control stem cell environments. So one of the things that we've been doing is to try to take stem cells and control their environment. So typically what people do when they want to take stem cells and make, let's say, heart cells out of them, then is that they put the cells in these non-adhesive dishes. So these are surfaces that the cells don't like to stick to. So what happens is that the cells start seeing each other and aggregating. And inside these aggregates of cells, you start getting all kinds of differentiation behavior. So because cells kind of think as if they're together, they're seeing each other, they feel good, then they start saying, okay, we need to start differentiating and make different kinds of tissues. So that's kind of what they see here. And what we want to do is control this kind of environment. So we can use these little wells that I told you about and control how many cells come together and form these little aggregates, and based on that try to see whether we can start differentiating them to different cell types. So one of the things that you can do is that you can control this aggregation process much more controllably inside these wells compared to this random heterogeneous processes that most people use. And then you can start asking questions about how the size of these aggregates actually affects the differentiation of the cells. Because of course you want to take the cells and you want to make the cell types that you want and not just get all kinds of random things come out of these differentiating cells. So we've been interested in asking questions. What happens if you have a small aggregate of cells? These are little aggregates. Literally here there's like dozens of cells that are compacted together. Compare these small aggregates to medium-sized and large aggregates and look at their differentiation into either heart cells or blood vessel cells. And really all this stuff is the experimental details, you don't need to worry about it. But we wanted to ask the question, does the number of cells, how many neighbors you have, affect what kind of cell you wind up becoming? And one of the things we saw earlier on was that, yes, there is a lot of effect on how many neighbors you have on what you wind up doing. So what we see here, let's look up here. So basically these small aggregates don't have too much of this cardiac or heart cells. The red things are basically indicative of heart cells. So whereas the medium-sized and larger aggregates have a lot more of these heart cells coming in them. So what this says is that the bigger the aggregate, then there's more of these heart cells developing. You can do a lot of other kinds of analysis, which is, if someone is interested, I can go over them. But basically, they all say the same thing. Larger aggregates make more heart cells. Now, if you look at the smaller aggregates, then you see the opposite response. You see that blood vessel cells actually come up more in these small aggregates. So then, if these stem cells have less neighbors, then they start becoming more blood vessel-like. So this is what one can look at here is this red. This red here now is blood vessel marker, and you see that these small aggregates have more blood vessel-like compared to larger aggregates. And if you zoom in and you look at these blood vessel structures, then you start seeing these capillary networks. So you start seeing structures that look like blood vessels inside the body, or very small capillaries inside the body. So this kind of stuff is good because now biologists can use it and start seeing how you can make more of these cells. And there's a lot of other kind of analysis that one can do, which I don't know it's really as exciting as to go over in a short amount of time. So what I'm going to talk about is how now you can do more complex stuff. So one of the things that you can do now, so what I just showed you is a phenomena, right? So you do something, something happens. But as scientists, you need to do more than that. You need to understand why things happen. And these kinds of controlled systems can also be used to assess the molecular mechanisms, the reasons why things happen. So, for example, here, these are all these names kind of may sound really weird, but what they are is that they're different proteins and different kind of signals that are there in the environment of the cell. And what you can see here is that if you basically these bars indicate basically the level of gene expression of these different things. So basically what it says is that the large aggregates here, and these are the small aggregates, And you see that in the large aggregates, in this case for example, this molecule is expressed a lot, whereas in small aggregates, you don't see it expressed. And alternatively, in this other case, the small aggregates express this other molecule, and the large aggregates don't express them. So you can start understanding the molecules that are involved, and then be able to use these molecules now in other systems to differentiate these stem cells. So this is the general kind of concept I want to